Hey there. Welcome to Chapter 44. This video will cover 44.1 to 44.3. And so let's get started talking about osmoregulation and excretory systems. So the unit of measurement that we use to measure concentration in osmoregulation is the osmol, which is 1 mole, which is a 1 molar solution, or 1 osmol. And so human blood, just to give a relative perspective, 300 milliosms or milliosmoles. The ocean, a bit saltier, is 1,000 milliosmoles. And so in the past, we used the words hypotonic, hypertonic, and isotonic. And those words tend to refer to cells and fluids moving into or out of cells, or solute moving in and out of cells. And the words that we're going to use right now are a little bit broader. So we're going to use hypoosmotic, hyperosmotic, and isoosmotic. They transfer very similarly, and I'm sure that you can wrap your head right around that. And so osmoregulation has evolved, and one thing to think about is that life on Earth exists in just every little niche. If there's a way for life to live in a certain environment, then it'll figure it out. Give it enough time. And so we have life living in salty environments and in freshwater environments and on land. And so when we have organisms living in, let's say, salt water, we're going to find two different types of organisms, an osmoconformer and an osmoregulator. So osmoconformers, they conform to their environment. And so they're going to maintain an internal environment that is isoosmotic to the environment, or that is the same. So if we were to look at this jellyfish here, the inside of this jellyfish is about 1,000 milliosms, which is the same as the surrounding environment, the ocean, 1,000 milliosms. So this is going to be found in organisms that are not very complex. Their organs function less effectively, but it takes less energy for them to survive because they just live in and exist in that osmolarity. So osmoregulation is the process of an organism regulating the concentration of solutes within its internal environment, essentially homeostasis. So if you were to jump into water and swim in the ocean, or if you were to eat something salty, your insides need to maintain a very specific salinity. And so you have organs, specifically your kidney, that are going to work to maintain or to make sure that your inside environment stays at that very specific salinity, or your skin. Your skin's going to keep the salt out as well. So there's a cost to this. It's energy. It takes a lot of energy to have kidneys. It takes a lot of energy to have skin that's impermeable to salt water. And so the benefit, though, is that we can live in environments that osmoconformers cannot. So we can live in saltwater environments, we can live on land, etc., etc. So we have now looking at two types of fish, saltwater and freshwater fish. And so if we're looking at a saltwater fish, we're going to try and understand its regulation. And so saltwater fish are going to be hypoosmotic to the ocean water. So that means there's going to be less salts or less solute on the inside and more on the outside. And so therefore, water will flow out of the fish because it's going to be through the process of osmosis. And so therefore, the fish is going to have to drink salty seawater. The water will leave through the gills and its skin, and its excretion will be high in salt. And so it has to drink the seawater in order to get the water, and because it is losing water at such a high rate. Now the opposite is going to be true for the freshwater fish. They gain water by osmosis. So there's more, they are hyperosmotic to the freshwater, and so therefore water is coming in from the freshwater lake, and so they're going to gain water through osmosis through their skin, and they're going to their excretion will be dilute because there's so much There's so much water coming in, and they're going to gain salt ions through their gills. So they're going to need to maintain and have salt because we all need salt to live. And so they're going to get those through their gills. All right. There are different types of osmoregulating strategies. Some are more complex than others. So let's start with this very basic concept, and it's very broad. And so if we look too closely at it, we're going to be able to find holes and it'll get confusing. So let's just paint a very broad picture to gain an understanding of what a transport epithelia is. And so here, if you have an organism and they have blood, which if they're a warm-blooded organism, they will, they're going to need to regulate the amount of salt and the amount of water that they're going to be losing through excretion. And so here is going to be this epithelial, this wall of cells. And that wall of cells needs to maintain a certain osmolarity to allow or to regulate how much water or how many solutes are going to be released through the secretory tubule, or for us, to lose it as urine. And so this will be the secretory tubule, and so we're going to lose the solutes or lose water through this, and then this is the waste. This is going to lead to the exit. So the way that we maintain solutes in this transport epithelia is that we have to use active transport in order to maintain solutes, to bring solutes in and to keep them there in the cells so that they can act as a regulator for how much salt and how much water can pass through. So that's a very broad concept. Let's look at an example of this. One is the albatross. The albatross is very cool, very big bird, 6-foot wingspan, and they can stay out at sea for long periods of time because they don't need to drink fresh water. They can drink the salty seawater, which is really cool. And so what they do is they, if you can picture this, they drink the salty seawater, it goes down their esophagus, into their stomach, and then it's going to be absorbed into their bloodstream. And so here is the blood vessels of the albatross. And so all of the blood vessels, or all of the, not the blood vessels, all of the blood in the albatross is going to eventually go through this gland, this nasal salt gland, which can be found right here and right here. And so here, this gland works with a transport epithelial layer to remove salt. And so the excess solutes in the blood will flow along the epithelial tissue, and the salt, the plasma ions are going to diffuse out in a very high rate because of the salinity of the membrane, because it's going to be regulated to allow lots of salt out. And so the solute is collected in the central ducts, and it actually, the bird doesn't dehydrate because it drips the very, very salty water out of its nose, which is really interesting, which is why it can drink the salty seawater and not dehydrate. If we drink the salty seawater, something very different happens, but we'll talk about that later. And I'm going to move me over here. So there are different systems that remove nitrogen because life on Earth exists in, like we said, every different niche. And so we need to adapt to those niches and those environments. And so excretory systems remove nitrogen because an abundance of nitrogen can be toxic. Why is it toxic? Because nitrogen can form ammonia, and ammonia is a toxic substance, and it needs to be removed. And so there are different methods of removing removing this ammonia. And so if an organism is excreting pure ammonia, it's probably living in the water. It can just diffuse right out. The second that it is present in the organism, it can diffuse out. And so you don't need a system to transport it out, and you don't need to dilute it. It's going to just be dilute enough that it'll just pass through, and easy peasy. The next, if we're living on land, we can't just diffuse our ammonia out. We need to transport it out of our body, and that's done through our ureters. That happens in the kidneys, and it passes through our ureters and sits in our urinary bladder until we excrete it out. And so we need to make that ammonia less toxic, so we dilute it with water. And so therefore we can pass it without getting sick. And so that is what urea is. It's ammonia in water. And so that is what a lot of land animals will do, is they will mix their ammonia with water. Another adaptation is uric acid, also known as guano. This is a white paste that I'm sure you've heard that bats have guano. It is chemically altered. It is relatively non-toxic because the ammonia is chemically altered into just a different substance. The organism is not going to lose a lot of water, so that's a benefit. However, it does cost a lot of energy to produce this guano or this uric acid. And so we move on to different excretory systems that remove nitrogen. If you've ever seen the evolution documentary with Neil Shubin, he has a very interesting way of showing that life uses the same systems over and over again. They call it tinkering with the same basic plan. And so we see the same things, let me move myself. We see the same ideas, but just looking a little bit different. And so one way we see it is in this protonephridia. They use those flame bulbs, which is just a very simplified method. The metanephridia, which are earthworms, and they are going to use capillary networks, slightly more complex. And most vertebrates, birds, reptiles, mammals, we all have kidneys. And so our kidneys are going to work to produce excretion, but also osmoregulation, meaning it's going to help us maintain our water so that we don't lose all of our water while trying to get rid of our salts and the ammonia. And so that is the way that the kidneys work is this very basic plan. We're going to look at different steps: filtration, the filtrate, reabsorption, secretion, and excretion. And so here, this is when we're learning about the kidney, I want you to come back to this very basic concept, because it's nothing more than this. Sometimes it gets overwhelming, and if you get confused, just come back to this basic concept. There is a capillary or an artery that's going to pass into, at one point you're going to learn that's called a Bowman's capsule, but it was a Bowman's capsule. But right now we're going to just call that a filter. And so the artery goes into the filter. The pressure of the heartbeat pushing blood through the arteries is going to push some of this solution through the blood vessels. So it's very porous. The blood vessels are very porous right here. And that's going to create something that we call a filtrate, which is just filtered blood. And the large red blood cells are going to remain in the arteries. And so we have a yellowish plasma that is going to be traveling through this tube, this filtrate tube, and so, or tubule. And so it's going to go through a couple of different steps. Second is reabsorption. So when this filtering process happens, it's not very selective. And so it's going to push out things like glucose, things like amino acids, things that are important that we want to hang on to. And so the body is going to actively, or use ATP, pump those back into the capillaries. And then, so that we recapture all of the stuff that we need. And then there's going to be another step called secretion, where we actively pump other toxins into this tubule. And then finally, excretion. That is when it is leaving the body. And so that is it for this set of notes, and we will talk about the kidneys next.